Does anyone know where Oregon Health and Science University is? Does anyone watch Portlandia? So we're right in Portland, and we're actually on top of a big hill. And I took out the pictures in the interest of time, but it's actually a very beautiful place, and it's actually a very up-and-coming place as far as trying to understand neuroscience and the genetic basis of psychiatric and neurodevelopmental diseases. And what I'm going to talk to you today is basically the work that I've done for roughly the last 4 years or so that's really been focused on how we can try to start unlocking the genetic picture of autism. So you probably hear in the news a lot, there's now a new autism candidate gene. We found a new candidate gene. There's not a single gene that causes autism. But really, until the last few years, we've had a really hard time moving from kind of a handful of case reports and suggestions that this gene or pathway might be involved to actually moving to a situation where now we have genes that we really know and have strong suspicion are really important and really may underlie the biology of this complex disorder. And that's really what we've been trying to do because as was mentioned here at the Conte Center, we really want to try to model these things and understand how the biology is really working. And so today, I'm going to just briefly introduce you to some of kind of the challenges and goals of genomics in kind of this day and age, and how, in particular, we're trying to address these things in complex genetic diseases and disorders. And then I'll tell you about one approach we've taken to try to unlock this genetic architecture, and this is using a family-based or trio-based exome sequencing paradigm. And this basically allows us to identify candidate genes in an unbiased way. But that's really not good enough. And so we need a way to actually validate these are truly known genes. And I'll basically just give you a brief introduction to a new method we've developed that for very little money, we can actually resequence genes in many thousands of individuals so that we can try to replicate findings. And finally, depending on how much time we have left, I'll tell you just a few hints of what's coming in the near future as far as kind of autism-related research. So first, the goals. So what are we doing here? What are geneticists doing? So the real thing that you're trying to do as a geneticist is we're studying DNA for the most part. And as you probably know, DNA is really the blueprint for all biology. And it's a 4-letter alphabet, A's, T's, G's, and C's, and it's a language we don't speak, right? So we didn't grow up speaking DNA. And what we've been trying to do to actually learn how to speak this language, of course, is same as you would do trying to learn any language. So we're trying to read a lot of books, right? So we're trying to learn this language by sequencing lots and lots of DNA. And so kind of the first, I would say, kind of a monumental moment in DNA after the double helix was the draft version of the human genome. And this actually happened while I was still an undergrad and learning molecular biology. And this really, for the first time, gave us at least a draft of all of the genes in the human genome, roughly where they are, and started to give us some insights into human variation and human disease. And it's probably can't be overstated how much just having the rough blueprint of where the genes and the DNA were in the genome accelerated discovery on a whole wide range of topics. So, back in 2011, to sequence about 1 million base pairs of DNA, it would cost you about the same amount as this kind of beat-up used car here, right? So, almost $10,000 to get 1 million base pairs. And really, in the last 7 or so years, the price to actually do this has dropped by several orders of magnitude. So in 2007, even before iPads existed, for about $500 now, you could actually sequence 1 million bases of DNA. In 2009, this dropped to rather around $1, so about the price of a Snickers bar. And today, you can do this for about a dime. And there was some big excitement a couple weeks ago when one of the main new sequencing companies announced kind of this new version of a sequencer that potentially can get us to the point where for about $1,000 you can sequence a human genome. And so what's kind of led to this revolution? And it's really this idea of doing things on a massively parallel scale, so massively parallel sequencing. So I'm not going to go through the exact details of how this works, but if you learned anything about kind of Sanger, traditional Sanger DNA sequencing, you probably know that it was kind of a base by base sequencing readout. Initially ran on gels, then we moved things to running capillaries with fluorescent tags, but all of this was basically done one molecule at a time, reading one base at a time. And Jay Shinduri was who I worked with my postdoc with, was one of the first people that actually, instead of doing this thing one at a time, why don't we try to sequence thousands and now millions of these strands of DNA all at once? And so this is done roughly on the same thing as a kind of slide that you would look at fixed tissues. So it's just on a little slide, but has millions of these little dots, and each color will represent one base that's getting sequenced for each one of these dots. And so the basic paradigm of DNA sequencing is actually fairly simple. So we have some DNA molecule here, and the different colors represent different bases. We shred it up into little pieces. And then these little pieces can then be read by some sequencing technology. And as I mentioned, going to kind of this massively parallel way of doing things has dramatically reduced the cost of doing this. So then the final kind of challenge of the paradigm is you have all these short stretches of DNA, and now we have to figure out how they actually stitch back together to represent kind of this original precursor molecule. In an ideal world, you would just sequence this molecule because you would preserve all of that information about the structure and the order of the DNA. And so really having the human genome has allowed us to take some shortcuts because we can align these short fragments to that reference genome. And that is a much simpler proposition than trying to reconstruct this molecule kind of from scratch. And so as Barry alluded to, really there's a lot of excitement about pushing the envelope of medicine into something called genomics medicine or personalized genomic medicine or precision medicine. It kind of depends on what your favorite buzzword is. And I would say that we're still several years away from actually making this happen on a kind of a large scale, but there are already good examples of where this is actually happening in both Mendelian disorders, but also in cancer biology. And so the idea is pretty simple, and this is really the kind of the holy grail of genetics is, you have some disorder or disease that has a genetic risk, and we want to basically understand what are all of the genes and pathways that are affected in this disorder, and use that to think about the biology and start to model the biology so that we can actually develop novel treatments. And then finally, we want to make that personal. So we want to know what your genotype is so that we can tailor the treatment for you based on that. And I've highlighted this article here that was a drug trial that was published in the New England Journal of Medicine. And this was on a gene that causes cystic fibrosis. And cystic fibrosis was actually the first gene ever, what they used to call cloned in humans. So this was before we had a draft of the human genome, and way back in 1989. And about 20 years later, we had the first drug for the first genotype, which is this G551D mutation that affects about 3% of kids with cystic fibrosis. So it's been a long road from that initial gene discovery to having drug treatments, but we're really excited about the potential of new technology to rapidly accelerate our advances in this kind of way. And so if you look at how we've been doing as human geneticists as far as trying to understand genetic diseases and disorders, you can kind of break things broadly into two classes. So disorders that are Mendelian or follow the Mendelian laws of inheritance that you've probably learned about in your courses, and things that are more complex. And complex is really catch-all for more or less the stuff that we don't understand. So pretty much anything that does not follow kind of the traditional Mendelian laws gets grouped into complex. And I've just listed some of the Mendelian disorders that have been solved here. I already mentioned cystic fibrosis, which was the first one. But as you probably know, these kind of break down into recessive and dominant and also sex-linked traits. And there's actually a major effort going on right now in the US to basically solve the other half of these Mendelian disorders in the next few years. So roughly, most of Mendelian disorders will hopefully be explained in the not too distant future. But for complex, we've had a much tougher time. And that's really because for Mendelian disorders, largely these are things that are caused by a single gene. And the same gene is mutated over and over again in independent families. And so that makes our job much easier because geneticists are really great at counting to 1, but anytime we have to count above 1, we have a really hard time. And so that's really why for complex disorders, such as things like autism and epilepsy and schizophrenia, we've had a much tougher time. And so I'm not going to spend a ton of time talking about the phenotype of autism. We could probably have a whole talk about that. But I just wanted to mention broadly what the previous and current diagnosis criteria are. And so until last year, autism was basically diagnosed by questionnaire and observation by having kind of pervasive impairments in 3 overlapping domains. So social interaction, language and communication, and having restricted interests or repetitive behaviors. So if you had all 3 of these things in a constellation, you would get an autism diagnosis. If you had 2 of the 3, you would get a diagnosis somewhere on what's more commonly termed now the autism spectrum. Just this year, the kind of diagnostic manual used by psychiatrists has been updated to what's called DSM-5, and now the definition is now these kind of 2 domains. So if you have social communication and interaction impairments and restricted interest and repetitive behaviors, now you get an autism spectrum diagnosis. And it's still a big area of research on how kind of this changing definition is going to affect both kind of access to services, but also our research. So importantly, there's no medical test. You can't go to the doctor and get a blood test for autism. And the prevalence estimates have continued to go up in recent years. So just a few days ago, the CDC released their new report that suggests on average, somewhere around 1 in 68 children in the US have an autism spectrum diagnosis. And I should just point out that there's very large confidence intervals on this rate. So even in the US, comparing across different states, the prevalence estimates may range from 1 in 50 to 1 in 200. And so I think you have to interpret these results with some caution, given that in the same country, using the same study and the same design, we see such variability. But it's definitely an active area of research that people are trying to get a handle on. It's been known for quite some time that autism more commonly affects boys than girls. And we also know that these kids and eventually adults often have overlaps of other complications. So about half also have intellectual disability. About 30% have some risk of seizure in their lifetime. And having a child with autism, the CDC's estimated costs a family economically somewhere around $4,000 to $6,000 kind of extra care from a variety of sources per year. So we've known for a long time that autism likely has a very high heritability. There's been some more recent estimates that suggest that it may be around 60%. The earlier studies suggested maybe around 90%. But in any case, this is still a major contributor to the risk. And as a geneticist, we're really trying to get a handle on this genetic components and eventually, hopefully, use that information to start to get at the kind of the larger picture and the more of the nurture component. So importantly, there is no such thing as a single gene for autism. That's been pretty well demonstrated over the last 20 years of research. And so this kind of has led to this realization that autism is a very what we call a genetically heterogeneous disorder, which just basically means there's many, many genes, probably hundreds of genes that contribute in the broad population to developing what we call the autism spectrum. So really, we really want to understand the story of autism. These are all kind of the classic parts of a story. And as a geneticist, I'm really interested in this question of who are the genes. And I think as a geneticist, I would argue that without knowing the genes, we really can't tell the rest of the story. We can't understand really what the biologic basis of autism is, what the real underlying phenotype is on a biological, physiological level. And we want to know who the genes are because we want to know kind of when these genes and then their eventual proteins are active in development, what parts of the cell do they normally function in, so that we can try to understand kind of where to model things and where to develop treatments. And finally, the why is because for especially kids on the very low end of the spectrum, we want to find some way of making their lives better. And so the other reason I really like genetics is it's a knowledge-free approach. We don't have to know anything about biology. We just have to know about genetics. Really, this is, when I was a student, really got me excited because this was really the one field in biology where you could really discover novel things that no one would have ever predicted. And so this is the quote on the way into my office that I see every day. And I think it's really great to keep in mind, especially as we start to think we know everything. The obstacle to discovery is the illusion of knowledge. So we really like genetics and also using genome-wide approaches because they can lead to kind of the most unexpected novel discoveries. So if you kind of look at how we were doing as far as understanding the genetic architecture of autism, kind of circa 2011, it's kind of broadly shown here. So for about 70% of the kids, we really have no idea what their genetic basis or liability might be. There had been a lot of interesting findings in the 3 or 4 years before 2011 that had shown that copy number variants, so these are large deletions and duplications of many tens to hundreds of genes, play a very important role in the kind of the genetic risk architecture, and have really kind of led to a better understanding of what the kind of the genetic risk factors might be. But unfortunately, since many hundreds of genes are often deleted, it doesn't really pinpoint exactly what genes we need to model and what particular parts of biology are affected. There has been a lot of interest now in these single gene or metabolic disorders, which are sometimes called syndromic autisms. And especially in the kind of the recent scientific literature and popular press, a lot of things that are very rare syndromes that 20 years ago no one would have ever grouped with autism, because of this kind of shared symptomatology, are now kind of getting grouped in with autism. And they've actually led to some important insights into the potential biology as well. But going to University of Washington for my postdoc, we really wanted to try to get at this 70%. And to do this, we really needed new approaches. So kind of all the standard human genetic approaches were failing for the last 20 years or so. And so we really wanted to do something different. And so what I went there to set out to do was to study families that look like this. And we call these simplex families or sporadic families. And these is an example pedigree of one family. Fathers or males are shown with squares and mothers are shown with circles. And my diamond here are just kids of miscellaneous sex. If you have a filled circle or diamond or square, then you're an affected child. So so basically in these families, we have the appearance of a child who's affected with autism that has occurred kind of out of the blue. So there's never been a previous family history of autism in the mother or the father, any other children, or any extended relatives. And so the thought was that, well, at least in some of these families, potentially new mutations that just happened when this individual was conceived may be playing a role. And we call these new mutations de novo, which is just a fancy science way of saying new. And we really wanted to look at point mutations, so single or small number of base changes, because these would actually pinpoint down to individual genes and give us kind of the granularity that we haven't had previously with whole genome approaches. So importantly, if we just focus in on point mutations and we could actually identify them, we only expect about 1 per generation to occur in just the coding regions of the genome, which is about 1%. And then we thought, well, if we could do this, we could take advantage of a statistical property that you'd expect that most mutations would occur randomly. And so if that's your assumption, then it should be unlikely that you find mutations at the same gene again and again by chance. And so if we found the complement of that, if we found genes that were recurrently disrupted over and over again in kids with autism that were not mutated in other individuals, that would suggest that those might be kind of bona fide genes that are related to the disorder. And so how do we actually get at those point mutations? So that's what I'm going to describe to you next, which is this trio-based exome sequencing paradigm. And so back in 2009, the word exome did not exist. So this is a very new field. You may have noticed that ohm gets tagged on to a lot of stuff these days since the genome was sequenced. And the exome is really simply all of just the small protein coding regions of the genome. So all of the kind of the triplet codons of the genome. And Sarah Ang actually developed this approach as a first year graduate student in Jay Shanduri's group, and basically showed that by sequencing the exome of kids with Mendelian disorders, you could actually identify in a very fast way kind of the causative gene for these more simple disorders. And so, you might be asking, well, what's the big deal? Why has it been so hard to sequence just the exons? If the exons are interesting, and that's what codes for proteins. And really the challenge is that the genome is not ordered nicely, like words in a story. And so exons are shown here in this kind of graph of a gene from the UCSC genome browser as these kind of fat boxes. And so these are the little boxes that actually code for proteins. And interspaced between them are these intronic sequences that play other important roles but don't directly contribute to the sequence of the protein. And so there's somewhere around 250,000 of these exons that are scattered throughout the genome. And so you really need a new way of trying to enrich for just that protein coding part of the genome, at least until we get to kind of the next order of magnitude decrease in sequencing costs. And so the solution that came about in just 2009 was to do something like this. And I'll just briefly walk you through it. So you start with your DNA. And as I mentioned, you just shred it up into small little bits, which are shown here. And some new technology allowed us to actually design probes that were complementary to all of those little exons. So all those 200,000 exons scattered throughout the genome. And some of the commercial platforms, we actually had somewhere around 2.1 million of these probes. And each one of these probes has a little bit of a biotin on the end. And if you do your experiment correctly, you can hybridize these probes to their complementary short stretches from the genome. And then using a kind of a bead that will bind biotin, you can pull down selectively all the little bits of the genome that correspond to the exons, and then just elute these off of some beads. And this gives you an enrichment of the kind of the exome part of the genome. And so then you can basically just sequence these things on the kind of massively parallel sequencers. We set out to do this in a first set of 20 pilot trios. And what also made this possible for really the first time was a major effort by the Simons Simplex collection, which was funded by the Simons Foundation, to set out to actually collect a large number of these families that had the same type of presentation where you just had a single affected individual. And over 5 years or so, they collected over 2,700 of these families. And so now, our challenge after we actually sequence the exome is how are we going to find that one new de novo mutation kind of in the background of 20,000 or more variants. And importantly, kind of the more likely something is to be, the less likely something is to be true, the more likely what you find is going to be false. And so this is kind of a common reality across everything. And so since our probability of being correct that this is really a new mutation is so low, it's likely that most of the things that we think are de novo mutations are actually just kind of red herrings and artifacts. And so we came up with a pretty simple strategy as far as how to filter through this signal-to-noise problem. And I'll just briefly walk you through it here. So we sequenced the exome of these 3 individuals, the father, the mother, the child. We call all the variants by comparing the sequences to the human reference genome. And then we ask, well, which of these variants are present in the child and not present in the parents? So they basically violate Mendel's laws. And you get somewhere around 50 raw de novos. And I told you that you only expect about 1 of those to be true. And we came to the realization that since we were sequencing many thousands of other exomes for other reasons, most of these would probably be either systematic artifacts, so they're errors that occur over and over again from either the way that we're doing exome sequencing or the way we're analyzing the data. But a second major class may be just things that are common variation that for one reason or another we just didn't sample very well in one of the 2 parents. And so we screened these 50 or so raw calls against about 1,000 other exomes. And that's actually a very powerful filter. So it gets us down to roughly 2 de novo mutations per child. And that's very easy and tractable to go in and do a little more manual curation and look for some other potential artifacts. And then we actually did kind of forward validation of these events using kind of the traditional Sanger sequencing method. And we're able to confirm somewhere around 1.3 new mutations per child per generation. And so in just those first 20 pilot families we did, we actually found 4 mutations that we thought were potentially strong candidate genes for being involved with autism, suggesting this might be a very high yield approach at identifying novel candidate genes. So with some generous funding from the Simons Foundation, in basically 1 year, we were able to go from 20 trios to 200, which is a pretty remarkable thing to think about from kind of where the field was when I was a graduate student to where it went as a postdoc. And so we also sequenced, so in total we had about 209 trios, but we also sequenced 50 unaffected siblings, which forms what we call a quad. And so this was over 670 exomes. And importantly, this is billions of bases that we actually genotyped, which is pretty phenomenal. And so really for the first time, we could, in an unbiased way, start to look at what are the different properties of human mutations, even regardless to whether they have anything to do with autism. But also, of course, we want to see, can we identify any interesting candidate genes from this approach? And so I'll just briefly summarize for you some of the kind of mutation properties we identified. So in these 209 probands, we had 260 de novo mutations, and that breaks down to about 1.3 per child. And most of these are actually predicted to change the protein structure. And that's actually pretty close in line with what you'd expect by chance. So if you have learned kind of the codon tables at some point in your life, or you will in the future, you'll realize that most of the codons do not lead to a truncation. There's only 3 out of 64. The truncated proteins do, and very few, maybe around 20%, lead to a synonymous change. So roughly about 75% of your protein, of your random mutations should be protein altering. So as I mentioned in the introduction, we want to use recurrence to firmly implicate new genes. And we had a little bit of recurrence in our study. We had 2 loss-of-function mutations in a gene called CHD8, which is thought to have some interaction with the chromatin and DNA binding. So the chromatin is a protein structure that wraps DNA in the nucleus and regulates how it's expressed. We also had 2 mutations in a gene called netrin G1, which may somehow be involved in kind of axon patterning in neurons. So this was one result that got a lot of news in the popular press. And so it had been predicted for a long time that mutations should have a bias for coming from the father's sperm. And we were actually able to demonstrate that in an unbiased way. And so of the 200 or so mutations, we were actually able to determine which parent's genotype or haplotype they were inherited on, or they were formed on. And so 42 out of 51 came from the father, and there was actually a positive correlation with paternal age. And some nice genome work that came out after this has shown that there's roughly about a doubling of the father's mutation rate every 16 and a half years or so. And so 2012 was really kind of a landmark year for autism exome studies. So these 4 papers were published, 3 of them back-to-back in Nature, and 1 published a few months later in Neuron. And even though we didn't have a lot of recurrence, you might think, well, if we put all these studies together, maybe we'll start seeing a better picture of things. And if you do that, we have somewhere around 900 or so families that have been sequenced, but we still have very few recurrences. So there's 6 genes which had 2 or more of kind of the most severe mutations that we identified. And you can do some modeling based on this recurrence rate, and it suggests, as I alluded to, that probably hundreds of genes would be involved under kind of a very strict, all diseases, kind of sporadic new mutations, and caused by in this type of disorder. And so we tried to look at other things that might give us some insights other than recurrence. And one of them was, does it look like these genes talk to each other? Are they part of similar networks? So I'm showing here just a circular version of a network where each one of these colored circles is a node. And this is very similar to, you think of your social network. So maybe all of you are various colored circles here, and these connections are whoever are the individuals maybe you're sitting next to, or that you have in your kind of Facebook profile. And you really shouldn't expect, if we just have a random collection of mutations, to see any type of network connections. But we very surprisingly had about 40% of our mutations all falling in this network. Some of the work from the Cold Spring Harbor group, which is the Eisenhoff paper, showed that there's also enrichment of kind of targets of a gene that has been associated with Fragile X mental retardation. So the other piece of evidence that we had was a qualitative difference. So there's not really a large overall mutation rate difference between the affected children and their unaffected siblings. But when you look at the qualitative impact of those mutations, which I'm just highlighting here, these are the nonsense and splice mutations, so the most disruptive we have in the cohort. So in this case, somewhere between 1.5 to 2 times the number of events are the kind of the most deleterious in the probands versus their unaffected siblings, suggesting that that there's an enrichment of more impactful mutations in the kids who are affected versus their siblings. It doesn't tell us which of those mutations are important, though. Basically, in summary, we find about 1.3 new mutations per child, and we are now at the point of trying to validate these candidate genes. So we have many hundreds of genes that have been identified from these thousands or so, but we really want to move to getting to known genes, because we want to spend a lot of time, talent, and treasure to actually figure out what these are doing. And so I'm running a little bit long on time, so I'll just basically give you kind of the very brief version of this. So you can imagine now moving forward, maybe you want to sequence more exomes, but maybe you have a lot of great candidate genes, and now you just want to start to validate these. in large numbers of individuals. But until recently, we really haven't had cost-effective methods to screen large cohorts. We want to screen tens of thousands of families so we can find recurrent mutations and use that information to implicate particular genes. And so I developed a way of doing this with molecular inversion probes. And I will point you to Wikipedia that has a very nice description of them and how they work. But basically, in a nutshell, very similar to the whole exome capture approach, but in a much cheaper manner, we can capture thousands of exons in a single tube and do it for less than $1 per gene per sample. And so we did this with 44 of those genes that we initially identified in those 200 families. And we actually screened almost 2,500 children in a matter of months. So this was over 100,000 gene resequencing experiments, which is a couple orders of magnitude more than was possible even a few years before. And so simply we could find all the rare variants in those families and try to see, did we find any that are actually similar to the exome study de novo, and find recurrences. So when we did this, we actually found 27 new mutations that were all de novo, that hadn't been previously identified. And they had a couple interesting characteristics that I'll point out to you. So one is that over half of them were actually very severe mutations. And this is much more than you'd expect by chance. So this would suggest that there's a very heavy skewing towards kind of severe loss of function mutations. Importantly, we had 7 mutations in CHD8, which was the gene that we had previously had 2 mutations in, and found other things that we had missed. And so how likely is it that now we have a real autism gene versus just kind of random chance? And so this really gets into kind of some probability areas. And I'm showing here some kind of toy probability distributions that are roughly equivalent to kind of the Poisson distribution. But you can imagine if gene A is CHD8 and we have 7 mutations, maybe this is just the most mutatable gene in the genome. So in this toy example, 10% of the time we'd expect to see 3 mutations. But if it's potentially one of these other genes that has a lower predicted mutation rate, then we might actually have something real. And so we came up with a way of trying to model this using kind of a random, kind of analogous to a weighted roulette wheel, where every gene got a different weight based on how likely it was to mutate. And then we could actually ask the question now, is 27 a lot of mutations to actually find? And so what I'm showing you here is just a graph of the -10 log p-values. And I'll skip ahead to show you kind of what the significance threshold should be, kind of correcting for all of the different tests we're doing. And basically, we never found 27 mutations in our simulation, and could show that these 6 genes that are highlighted here actually explain about 1% of autism. And these genes all function in novel pathways that we never would have predicted ahead of time had anything to do with these disorders. So just in summary, I've shown you how we've used kind of this trio-based exome approach to identify candidate genes, and how we've used targeted resequencing to actually validate these. And I'll just quickly tell you what we're doing next. So we want to see, can we push this paradigm further? Can we get all of the major genes in autism? And as part of this effort, we're sequencing in collaboration all of the families that are part of the Simon Simplex collection. And this is going to basically be about 2,500 families that are going to be available very soon, and will be a great resource to try to tease apart the rest of the genetics of the disorder. And then we're starting to get to the rest of the story. So I'm just showing you here 2 pictures of 2 of the kids who have CHD8 mutations. And I'll just point out that they both have kind of the same feature of a very large forehead and kind of ears that stick out. And we really think that by looking at more genetically distinct subgroups of autism, we'll start to maybe pull out some of these subtle phenotypes that may be very important for the clinical care. And finally, the kind of the when and the where part of the story is starting to take shape. So a lot of efforts are going on right now to now look at where these genes that we've identified function in biology, So there's a lot of interest in the synapse and the connection between neurons and autism for a long time. But this kind of novel, this genetic approach has led to kind of identification of novel pathways, so potentially the regulation of chromatin structure. And so there's a lot of efforts to try to understand that. And we have a lot of emerging data, genomics data on kind of when and where genes are expressed in development. And that's actually pinpointing potentially kind of critical periods in human development where these genes might be playing a significant role. So with that, I will put up my acknowledgement slides and thank, most importantly, the patients and their families that contributed to the work. Thank you. No, and that's really something that we don't have a great sense of why that difference is. So there's other genetic disorders that we know have a sex bias, and some of them have been mapped to genes that have nothing to do with sex chromosomes or anything else. And it's really unknown, is there a different biology in males versus females, or is it ascertainment differences? Is it a different presentation? And so there's active areas of research kind of in all of those veins to try to tease that apart.
The part where you're talking about the similar features, it seems like that would be something that people would look at first. It would be so obvious. Is there a reason why it seems like it's coming later?
So the two features I pointed out are pretty subtle and would not kind of rise to presentation from kind of a normal pediatrician visit or even a genetic counseling visit. So, for a large number, especially Mendelian disorders, that's exactly how they were identified initially, by kind of shared dysmorphic features and shared phenotypes. And people have tried to do the same thing with these more complex disorders with kind of little success, I would say. So. There's just too complex, there's too many genes involved. Yeah.
Thank you very much.
Thanks.